Hello everyone and a happy new year. Welcome back to hopefully 2021 is going to be a lot less crazy, but it looks like we're still doing this. So it's New Year's Day and I know there's been a bit of a gap in this series. We're going to try and get the respiration topic finished as soon as we can today so we can move on to other things. Okay, so last thing we were talking about was glycolysis, the link reaction and the Krebs cycle, which were basically mechanisms of very gradually breaking down glucose to release different high energy carriers, your NAD and your FAD with the H's crammed on the end, so reduced carriers. This is where the payoff comes. Okay, this is where we get our massive sort of electron transport party that gives us all of our ATP. Okay, so let's move straight on. So there's three ways of making ATP primarily, and these ways that you really should know. We've already discussed in the previous topic photophosphorylation, which is essentially using the energy from light in photosynthesis in the thylakoids there to create a proton gradient and generate that movement of ATP synthase. Okay, we're going to come back around to that protein again today. Substrate level phosphorylation is essentially cramming phosphate onto ADP using the energy from certain substrates, right? Certain specific reactions. So the phosphate is added directly to ADP. From the energy released from the substrate. So, for example, that happens in the cytoplasm in glycolysis, and it happens in the Krebs cycle. That's where we've generated some ATP in the first place. And then what we're going to be talking about today is oxidative phosphorylation. This is essentially where the ATP is generated. Right, the ADP is phosphorylated indirectly using energy from those reduced coenzymes we've been manufacturing in the last few stages. So our NAD and our FAD in their reduced form can release energy to build a proton gradient, and that drives ATP synthase. Okay, and that is what the electron transport chain really is. Okay, it's a means of generating a proton gradient to drive ATP synthase. So, moving on. We're going to do this in three levels again, sort of your basic, what's good enough for A level, and then take it up a couple of notches, right? So really what you need to know is this is happening on the inner mitochondrial membrane. Specifically, there's a difference across a membrane, and that is crucial, okay? As you can see over here, we've got the intermembrane space. So this is inside the first mitochondrial membrane, but not inside the second. And then we've got the mitochondrial matrix, the very center of the mitochondria here. Now, if we look, these two here, NADH and FADH, are what we've manufactured in glycolysis and in the link reaction Krebs cycle, okay? They are now coming in here, and this is where we get the payoff. So the first thing that happens is our NADH, okay, binds onto this first protein carrier that's wedged in this membrane, okay? In that membrane, right, you'll find that protein, and that protein both breaks down, both oxidizes the NADH back to NAD, and coupled with that, it pumps protons. Okay, so that energy from that breaking down is pumping protons out of the matrix and into the intermembrane space. So you're ending up with more protons out here, high concentration, and a low concentration in here. Okay, what you then end up with, obviously, left over is an electron. Okay, and that electron is going to, just like in photosynthesis, transfer energy from one protein to another. Okay, now this is a simplification. There are some bits missing here, and it might look different. This is taken from our A-level textbook, the most generic A-level textbook that most of you will have seen, but it does miss a few proteins out. So, for example, FADH here is just shown as breaking down into FAD and just pointing at an electron. There is a bit missed out there. We will come back to that in a little while. So this electron picks up some energy from its NADH and then from its FADH and gets passed down through these protein carriers, okay, and effectively that is all the pumping that's going to build that proton gradient. When it gets to this last protein here, it's combined with oxygen, which, if you've been paying attention, is the only ingredient in the GCSE respiration equation that we haven't yet talked about. Okay? So this is basically the last protein, and oxygen comes along, binds with those electrons, binds with some protons to form water. Okay, and that is also in the GCSE understanding of what respiration is. Okay, so we get this pumping going on. Oxygen is what we call our terminal electron acceptor. Okay, it takes that electron out, which is massively important because it allows more electrons to come down this electron transport chain, being passed from protein to protein, pumping those protons into the intermembrane space. Okay, this FAD now that we've manufactured, an NAD reoxidized, goes back round and goes back to glycolysis, back to the link reaction of the Krebs cycle to pick up more of the breakdown products from the new glucose coming through respiration. Okay, so it has to be recycled. Okay, and that's important when we talk about anaerobic respiration. Okay, so now we've got our proton gradient, and that proton gradient is a source of energy, and that's really important as well because this protein comes back here, ATP synthase. An ATP synthase is basically this water wheel type arrangement, this kind of combination of a helter-skelter and a water wheel through which the protons fall. Okay, they can't pass through the membrane because, as you'll remember from AS biology, this space in here is a non-polar region. It's a phospholipid bilayer, so the protons can't just force their way through. Their only way back through to even up this gradient is through ATP synthase. And as they pass through there, they spin the ATP synthase, and what that does is it causes changes in the protein structure that take an ADP and a PI and physically cram them together. So we're basically oxidizing these carriers, okay, to build a proton gradient which turns ATP synthase, hence oxidative phosphorylation. Okay, so that's sort of our textbook version there. If we take it up one little notch, okay, you can see this is the same thing here, but this actually includes this protein, this other protein that wasn't included in the previous picture, okay, where FAD binds on. Now it should say to FAD, not NAD there, that's a bit of a typo. But this actually is a mobile, it's the first what we call the mobile carriers. Now you wouldn't have to talk about mobile carriers in an A-level answer, but they do exist. The electrons don't magically just hop from one protein to another. There are actually proteins in there that take them across. Okay, so in this one we've got the same thing here. Look, we've got the NADH breaking down to NAD and protons, proton pumping, right, coupled with that electron moving down the electron transport chain. We've got our oxygen there as our terminal electron acceptor, okay, and then this phosphorylation of ADP using the power of that gradient. Okay, that's the sort of second level, which acknowledges that there's another protein there. These proteins do all have names. You don't have to know the names for A level. You really should for first year degree, of course. They do actually have names. So unsurprisingly, the first one is called NADH reductase. Okay, you can see why. Then there's this mobile carrier called ubiquinone in the middle, cytochrome reductase, cytochrome C, and then cytochrome oxidase. Okay, and that actually acknowledges the existence of these carrier proteins in here. Now, the only other place you might come across these carrier proteins is as part of questions on genetics. Because these ingredients are so important and so ubiquitous to all of life, often the genes that code for these ones, ubiquinone and cytochrome C, are used to compare different species. Okay, you don't have to know about them for ATP generation purposes for A level. You may have come across them in other exam questions, depending on your board and your spec and how diligent you've been in looking past papers. Okay, so that's sort of our third level up, but it's still showing us the same thing. We're using the energy from the carriers to build a proton gradient to power ATP synthase to make ATP. Okay, so you might also see diagrams drawn like this, okay, which basically show what we're trying to do here. That essentially this is gradual release of the energy in a form that can be turned into ATP. Okay, and these diagrams often confuse people because this y-axis here, look, be very careful, is the energy level, not physical space. So sometimes you see these overlaid onto the previous diagrams, which show you sort of the energy level, and you've got to be very, very careful to look at what the y-axis is actually showing if you want to answer those questions. And the bottom one usually is time. So over time, our NADH breaking down to NAD+, right, leads to a series of oxidation and reductions of the following proteins in the membrane, pump those protons, generate that ATP, and you end up with oxygen accepting those protons and turning into water, okay, metabolic water. Okay, right, so what we've said in these past sort of 3 videos, right, of all these different stages, that the breakdown of glucose provides energy to reduce NAD and FAD. They are our intermediates. The NAD and FAD that are reduced are used to create this proton gradient, and it's the proton gradient that powers ATP synthase and forms ATP. Okay? However, other substances can be used, other substrates can be used to make these intermediates, okay, to make this NADH and FADH. You don't have to just use glucose, okay? And those would be the fats and the proteins. So if we go back to this picture from the last video here, as you can see, look, we've got up here, if we look closely to the side there, look, you've got your triglycerides, your fats, okay? With the glycerol enters here, right, into the glycolysis stage, and your triglycerides, your fatty acids get broken down and enter here at the end of the link reaction as we go into the Krebs cycle, right? You've also got the same for various proteins that go in various places. So let's look at that in a bit more detail, okay? So this is how lipids are used in respiration. So you start with your lipid, there's your triglyceride, okay? Initially it's hydrolyzed, so these bonds here are broken, okay? The ester bonds you learnt about at AS level, right? Fatty acids and glycerol are formed. The fatty acids are broken down into 2 carbon fragments, and those 2 carbon fragments can replace the acetate that joins to coenzyme A to form acetyl coenzyme A. Okay, because a 2 carbon compound, much of a muchness, a lot of biochemical chicanery going on, can create acetyl CoA from fatty acids rather than from sugars. Okay, and the glycerol, well, that's a 3 carbon molecule. Right, what other 3 carbon molecule do we know about? Well, we know about triose phosphate. So these 3 carbons here can be phosphorylated, right, have phosphate added to them, become triose phosphate, and enter into glycolysis, turn that into pyruvate, and then through the link reaction. Okay, so that's how your fats can be used. Okay, proteins, all right, similar idea. Once you deaminate, all right, once you take away the amino group, the bit that this bit here, the bit that's ammonia, and that gets turned into urea. All right, that's in the thing called the ornithine cycle. That depending on your spec, you might cover, you might not. Okay, the rest of it is pretty much carbon, hydrogen, and oxygen. Okay, so really that's respiratory fuel. So if it's a 3 carbon, now depending on what the R group is, if there's 3 carbons, right, you end up turning that into pyruvate, and that goes into the link reaction. If it's a larger amino acid, 4 and 5 carbon compounds, well, it goes in there as Krebs cycle intermediates, so alpha-ketoglutarate, oxaloacetate, that kind of thing. Okay, so the point being that other respiratory substrates can be used, because really what's the point? What are we trying to build? We want that proton gradient, and the way we get that is through reduced NAD and reduced FAD. Okay, and it doesn't really matter how you come by that stuff as long as you can. So ATP synthase, right, massively important. We came across it in photosynthesis, we've come across it again in respiration, and it's so important. I want to finish today by showing you a particularly unusual video, right, that I was treated to in my first year of my degree. This came as part of a CD-ROM in the back of one of my first year degree books, and this was 2002, and we thought CD-ROMs were outdated then. So amongst all of the crazy diagrams and tables of information on that CD was this bizarre video where 4 Japanese biochemists are representing ATP synthase through the medium of interpretive dance. And you know it's got to be important if you can get biochemists doing interpretive dance. So if I can get the technology working properly, that's where I'm going to leave you today. Enjoy. And my favourite part of this, they gave themselves credits. Thanks very much. See you next time for anaerobic respiration.